Thank you, Dr. Davis, and thank you everyone for the opportunity to present here. So my name is Manish, and I'm a professor at Mount Sinai, where I direct a laboratory. I'm also vice chairman of the department. I want to share some conflicts of interest before I start, which is that I'm a founder of a small academic startup company as well called Linus Bio. All my conflicts are managed by Mount Sinai, and as I go through this, I will let you know when I'm presenting something from the company side, just to be extra careful about managing the conflicts of interest. I've seen the previous talks in the seminar series, and so I know that for many of you, the exposome concept is nothing new. But just to quickly recap, this is a relatively new paradigm that asks us not to study one environmental factor at a time, but rather the totality of all our exposures, the totality of our environment, whether it's social factors or toxic chemicals or something like that, and including lifestyle factors. It also asks us to focus not just on the external environment, but also what's inside your body, and including how our body responds to that. And really, the paradigm has arisen out of a big need that we recognize now that genomics alone cannot fully explain health and disease. Because if you look at the history of precision medicine, the past 20 odd years have really been gene centric, and we've realized that that alone is not going to move the needle enough. I want to focus on certain differences between genomics and exposomics, especially one that is what my team and I focus on, and that's time. Your genes are mostly static. If you look at your base genomic sequence, it's set at conception, and then sure, you have some epigenetic changes later on. But your exposome is changing at a rate that can extend from minutes to hours, but also months to years. And this constantly changing exposome is not something that we can uncover by just doing prospective cohort studies. I often get this from my colleagues who said, well, we're measuring time because we are doing a prospective study. However, that's not what I'm talking about. I'm not talking about snapshot measurements taken once a year because your cohort participants are visiting them and you do a blood test. Now, this idea has been put forth more recently. There was a very nice review by Dr. Aaron Haynes, who used to be at Cincinnati, is now at Kentucky. Also, my team wrote in the same journal issue. The idea was that studying time, the time dimension in detail, actually uncovers and allows us to look at patterns that otherwise would not be visible if we just use snapshot measurements. And this idea is not just something that we are promoting. There have been some very nice studies. For example, this paper in Science Advances talking about how, misalignment between your circadian clock and when you're taking in glucose-rich diet will actually create issues for you, for your health. And recently, I was honored to play a very small part in this trial, which was led by Konstantinos Makris, who's in Greece, where they actually did a randomized crossover trial looking at how contaminants from food are absorbed differently depending on the time when you consume it. So time matters, but time matters at a very fine scale. So start thinking in hours rather than just in years. I want to introduce a technology, and here's where I highlight my conflict of interest, because this technology was really developed with private sector money. You're not going to use NIH dollars to start building robotic platforms when you just don't have even a blueprint. So this is something that the private sector is very good at. And why did we build this technology? I'm just recapping what I said before, that we needed a way to measure hundreds of time points at an hourly interval rather than relying on just blood testing. And we obviously cannot take hundreds of sequential blood samples, even if we had microsampling things like pinpricks of your fingers and things like that. That just wouldn't work here. The idea is actually very, very simple. If anyone's ever seen a tree that has been cut down and there are growth rings in that tree, you're looking at a past history at an annual frequency. So each growth ring represents a year. And you're saying, okay, I'm going back in time year by year. It turns out that human scalp hair also grows in an incremental manner. You can almost imagine it having growth rings, and we are working at a resolution of about 1 hour because of the rate of the way hair grow, which is about roughly 1 centimeter a month. There are differences by gender, by ethnicity, and even within an individual, there are growth periods of rapid growth and slower growth. However, as a rule of thumb, you can think of hair growing roughly 1 centimeter per month. Now, how do we take advantage of this? So I'm going to show you on the right this graph which has these two black box labels. And imagine that in this person's history, we have taken two blood samples, one at 200 days and one roughly at 700 days. You're measuring a neuroactive compound. In this case, let's say it's tin. And most of you will know that tin is very neurotoxic, and we've studied it to some degree, and we know that it's bad for your mental health. If you looked at these blood tests, and I used to be a clinician many years ago, I would say, oh, well, exposure is not really a concern here because it just remains low on both tests. What I'm about to show you next is real data. We collected hair from this individual and we mapped it back almost 2 years, all from a single strand of hair. And we said, hey, let's build this on, in this case, it was about 2 to 3 hourly resolution. And here's what you see. There are periods of high exposure. It's just that the exposure happens, it comes down, there's some patterns in there of smaller peaks, and then again, it subsides. So snapshot measurements done once a year just cannot unravel these temporal dynamics that are so important to environmental medicine. Now, how do we do this? Because here are the challenges, right? Not all hair is straight. When I had hair, I don't have much left now, it was pretty curly. So when we analyze hair, we need it to be perfectly straight, and human hands just can't do it. We also need to open up the hair because the outside can be severely contaminated. If you're walking past someone who's smoking a cigarette, the nicotine residue will settle on your hair. It's actually very hard to just clean it out. So the biologically relevant information, the internal dose, in a manner of speaking, is in the core of the hair, not on the outer keratin layer. So this is how we unravel this problem over years of effort and lots of failures. We built this robotic platform. It takes hair, any kind of hair, curly hair, thin hair, thick hair, any kind of hair. It is so precise and it makes it perfectly straight. It just applies the right amount of tension to make it perfectly straight, and it stamps it on a slide, on a flat surface. What it does next is very hard to do with human hands. It actually opens up hair lengthwise. So again, I want to stress this point. We're not cutting hair cross-sectionally like with scissors. We're actually opening up this centimeter of hair along its length, and we don't have to use heat or anything like that. It's an entirely mechanical process. So if you wanted to imagine exactly how it's happening, think of like unraveling a Twizzler in a manner of speaking. Sorry, let me go back. I clicked the wrong button here. So as we open up this hair, we now expose all the molecular information inside. And then we take these special kind of lasers that release these molecules, and we have coupled our detectors, which are mass specs or mass spectrometers, to measure that hourly pattern. So from each roughly centimeter of hair, you're getting between 800 to 1,000 time points. And that's what gives you that hourly profile. It's like taking a blood test every hour. Now we use AI because we measure up to 10,000 molecular signatures. And so if you'd say 10,000 molecular signatures times 1,000 time points, that's a matrix of 10 million biochemical data points. We can't use traditional statistical methods. So I have a very smart team of 4 AI experts who take this information and then apply it, whether it's to exposure assessment or to a disease outcome. I just want to show you that the patterns we uncover are very much like this. In a sense, we are doing what your smartwatch or your smart ring does on the surface of your body. It measures 3-4 things, and then it derives multiple things like sleep quality from it. We are measuring up to 10,000 molecular signatures inside your body. It's like if you swallowed a wearable and it's constantly emitting this information. The great thing is you don't have to pay for this. Compliance is 100%. It's not like your smart ring If you were sweating one night, that ring will start giving you, different data, or the data might not be as accurate. So none of this suffers. Sort of biology has given us a variable that's in our body. One thing that I'm very passionate about, and I'll share my personal experience as a dad later in this presentation, is that we really need, as academics, to bridge this gap between research, because what we are doing is here, but the taxpayer is waiting for solutions. The taxpayer is saying, hey, where is the answer? It's been decades of academic research, but where can I go and buy this tool that helps me improve my environment? And I'm going to take you through that journey that I experienced, and why I founded a small company. So I woke up a few days ago, and I thought the sky in New York was pretty bad. And thank you, Canada, for this. But I looked at the news, and geez, Chicago was really hit hard by this. And if you look at the AQI index, Chicago actually ranked as having the worst air quality of any major city worldwide. I honestly have not seen an AQI of 701 ever, and I starts worrying when it starts crossing 100, and start thinking about wearing a mouth mask and keeping my kids indoors. But if you live in California and globally, these events are becoming more and more common. So we are seeing them almost every year now, and so this is no longer a rare occurrence. I was honored to help support the families who were affected by the Palisades fires, and I really have to acknowledge the families who have organized themselves. They're real champions. They put me to shame how actively they're involved in their community. So while I'm still helping with the current Canadian fires that are still ongoing, I don't have data there, but I would like to share data on this very relevant event. And I've seen previous talks in your seminar series on the fires, on various types of fires. So the Palisades fire started around January of 2025, and they were quite devastating, as you can see from these images. Now, as a researcher, here are the key problems, but these problems are really noted down by the community members that I'm working with. And again, I really cannot acknowledge them enough, the amount of effort they have placed in helping do this work. But for most of them, they have no idea what is background exposure. Like someone asked me today, well, the Canadian fire started about a week or so ago. I want a blood sample from 2 weeks ago so that I know what is your background exposure before the fires. Well, I don't have a blood sample biobanked anywhere. And that's true for almost everyone. We're measuring air pollution through, stationary ground-based sensors or satellite-based remote sensing, but that doesn't always reflect what is your internal exposure, how much is really in the body. The added problem arises when you are the owner of a house that has been exposed but hasn't been burned down. So if your house is completely burned down, you work towards building a new one. But if your house is still standing and it was very close to all the air pollution and all the pollution from the fires, then when is the cleaning effort enough? Is the house safe to come back? And you'd be surprised how difficult it is to come to that conclusion. And there's a whole different angle of dealing with insurance companies as well, because they have their own policies and how they operate. So this is where we came in. We said, hey, we have a technology that we can go back several months. We can tell you what your baseline exposure was using your hair, what was your exposure during the fire, and then let's track it forward as well, and to see, well, how much time does it take to return to baseline. Around that time, we were honored that the New York Times came to us, and they said, hey, we hear that you have a technology that can help us do this very deep investigative study. And this was a long, detailed study. I have a lot of respect for the journalists who did this. I was on a Zoom call helping this one family who were like the case study, and I was guiding the parents, the mother, to clip some hair off their child. So this image is taken from a Zoom call that I was participating in. And on the right, you actually see a hair strand. And the New York Times has taken a picture of a hair strand being analyzed in my lab, and we have dated it. And I'll go in later into the science of how we date it and and the error around that. So we showed here that the levels are high in these children. They've been exposed to various metal toxicants, and we have a California-wide data set that was funded as part of an NIH study. And on all of these metals, or a majority of these metals, they are high. And the pattern was such that when they came back home, the exposures would increase, and as they cleaned the house, the exposures would increase even more. When they left the home, the exposures were lower. And again, I'll show you some data. What followed next as a separate article, and something that is a real highlight of my career, was we realized that a California assembly member had introduced this bill and cited our technology. So it's not every day when you're helping people one-on-one, but also the work that you are doing is being introduced into the assembly of a state as an effort to change policy, an effort to say, look, insurance companies, we need science like this before we decide the house is safe and our citizens are safe to go back to their homes. Before I share this data, I just want to highlight this data is what I like to say fresh out of the oven. My team worked over the weekend because I was presenting today, and we analyzed all this data, and it was just released this morning. So it's preliminary. Please note that. It's not peer reviewed yet, but I do trust it enough to share it with you. It's a busy graph. It's not very aesthetic. Like I said, we just got it finished a few hours ago. So we haven't made it pretty by any means. But let me walk you through what we are measuring. When we analyze here, we have matrix match standards, which allow us to quantify the concentration. And we are doing this in a CLIA certified lab. What CLIA means is that a state agency has come and evaluated your lab for quality, and they have stringent quality control measures. We are also ISO certified, so that's an international level. So I'm saying that because I want to convey that this work is done in a very well organized and very good lab. I'm not presenting concentrations here. I'm presenting the levels of a metal relative to the internal standard in here, which is sulfur. So that's your intensity axis on the Y. On the horizontal axis, I have the time axis. Please know that these red dashed lines represent our estimate of where in the hair reflects when the fire started, but we have some error around it, how people cut their hair, variations in growth rates and such. So we put a pretty wide error band around it as well, because we can never be 100% sure that this path exactly reflects that one day. Now, I'm going to start off with something which might seem like a benign element, zinc, but in fires, remember, zinc is everywhere. It's in your household paint, it's in some of your piping, it's in automotive components and all sorts of things. So when a suburban fire burns, it's very different to a forest fire. You get exposed to a lot of heavy metals, and remember, you're breathing them in. Our biology is not designed to breathe in zinc, is designed to ingest zinc. Yes, you can eat a lot of zinc and still be okay, but inhaling is a whole different story. So just to help you, align yourself around this busy looking graph, this is the black line is the median of the Palisades community, but I'm also showing you in the gray all the data. This is just 9 samples, 9 people from the Palisades community who were exposed to the wildfire. We also worked with the community to get samples from that same time, that same community, but they were not exposed. They were living away during the wildfire. So it's the exact same community as a good control. However, because we have done large studies in California, when you match by age and things like that, we have another sample of 200, which is here in this pink line, that is from California, but this study was done well before the fire. So they are from the same geographic location, a different time. And then the yellow ones are actually from Sweden. So different time, but also a different geographic location. So we built it up stage-wise. You have the exposed group, you have a group from the same time, same geography, non-exposed. You have a group from same geography, different time, and then you have a group from different time and different geography. As you can see, when the fires start, there's this massive spike in an increase in the zinc uptake in hair. And this stays there, and then you see this gradual trend coming down. Of course, you have spikes and everything, but it takes over a year for them, or closer to a year and a half, to start returning to baseline measurement. There is this, internal dose that is constantly being exposed, and they're living in environments that take a long time to clean. Zinc might seem benign, even though I hope I've made a case that inhaling it can be harmful. Lead is, from whichever route, is not benign. And again, we see a similar pattern for lead. It goes up. The trend and the time it takes to really start coming down are different, as you would expect. But again, higher than any of the control groups by a lot. And then as the fire is in the past, things start declining. Same thing for nickel. And we have done many elements. A large number of them show this pattern, not all of them. Of course, we have this big spike here. I'm not going to delete data that is there. It's an outlier, but I've still left it in. So again, we see an increase in the uptake of many heavy metals, toxic ones, essential ones, but inhalation is not a good route for even the essential ones, that align with the fire and are always higher than no matter which control we use. And again, I just want to reiterate that this is preliminary non-peer-reviewed data. So that is my effort. And please, if you know anybody who's being affected by the current fires, or if you want to use this as a research tool, please reach out. We are very happy to help, because this tool, I think, is one of the few that can really help resolve this problem of acute disaster events. I want to start talking about something else, which I briefly mentioned earlier. We need to make the exposome more accessible and impactful. This cannot just remain as an academic science, and our approach cannot be just to impact policy. This has to be something that we can empower people with. And if you look at genomics, even now, precision medicine at many institutes really is genomic-centered precision medicine. So we have to enter into precision medicine, provide clinical tools. We have to have policy that can impact things at a global level, because we can change our environment. We have been successful doing it, whether you look at global lead pollution, air pollution, and many other factors. But unlike genomics, because you cannot change your genes, but you can change your environment, is something so many of us say repeatedly, we can empower people to improve their exposome. And that's something that I'm going to talk about very briefly. And what does that even mean? So put yourself in a consumer's chair. Put yourself in the average person who is not an environmental scientist. How many exposures are you measuring today in your own home? And for most people, the answer is 2. You're measuring temperature through your thermostat, and you're definitely measuring, because fire alarms are mandated, you're measuring fire or carbon monoxide, but that's not really continuous monitoring. It's just that it goes off when you're having a bad day. Maybe some of you are measuring a few other things like humidity, if you have a dehumidifier, or you do some regular radon tests, things like that. But really, you're really measuring very few. But we all know we get exposed to hundreds and hundreds of chemicals. So luckily, because I can tap into the private sector, we were able to say, hey, here is something that requires just a snip of hair with scissors. You can bring your children along. There's no painful, pesky blood test. There's no going to one of those labs that's half an hour drive away. Just clip some hair. You can do it while the kids are sleeping even. And we've developed this simple report. It gives you an overall score. It tells you all the different chemicals. We're also working on various organics and things like that. And then we can give you action steps. And these are simple. They are so simple, sometimes I would even call them boring. Like, hey, maybe start using a reverse osmosis filter. Mopping, how many of us know how to mop our house correctly? Wet mopping, microfiber versus regular dry mopping. How many of us or our neighbors would know that a vacuum cleaner with a HEPA filter makes a huge difference compared to a vacuum cleaner without a HEPA filter? So it's things like that, small tangible steps. Now, I would be a bad scientist if I just presented my own work. There are many other technologies out there, and you're all familiar with most of them, I imagine. Silicone wristbands, like Kim Anderson is doing this work. I've worked with Heather Stapleton at Duke, and she's doing an amazing job of developing new modifications to this. Urine sampling is ubiquitous. My colleague Itai Klug and Mayanji Chek Sade, they do satellite remote sensing, and they do it at a much higher resolution than I've seen done before. there is there are many different approaches to blood microsamplers, although I doubt if they can give you this continuous monitoring. But indoor air pollution monitors are available, and there are many reviews out there on good ones versus bad ones, and they can certainly help monitor your indoor pollution, especially at times like this. Like if I'm in New York and these Canadian fires are turning the sky orange, then I know some of that is coming into my house as well. What I'm going to do next is talk about one of the other buckets, and I'm doing this partly not just for scientific reasons, but partly as a parent. So I'll share my personal journey here. I'm the proud father of triplet daughters. They're 13, so I've got 3 teenage daughters at home. Yay for me. However, my youngest daughter by 1 minute is neurodiverse, and it's a real journey. And anyone who has a child with extra needs in the audience, you know what I'm talking about. One thing that shocked me, and the question I'll pose to you is, what is common amongst all these disorders? And as you think about that question, I'll give you my answer. My answer was that when my daughter was recognized as being neurodiverse and we were testing for autism and ADHD and lots of other conditions that we were concerned about, I realized 2 things. Genomics wasn't providing any answer. I would do a massive genomic screen, come home, and I would not know what to do differently tomorrow. All of us are walking around with some microdeletion here or there, but that doesn't really translate into a diagnosis or any measurable phenotypic change. So what's going on with my daughter? So environmental medicine, exposomics, to me was an area that could provide an answer where genomics couldn't. The question I asked you, what's common between all of them, my answer to what is common between all these conditions, my answer to that question is that none of them have a validated biochemical test. And that's what shocked me. Back in my clinical days, I was doing work, for example, in oral surgeries. I would have a massive path report. I would check for diabetes. I would check for bleeding disorders. You don't want the surgery to go well, but then the complications to impact the patient. When my daughter is being evaluated for autism, I'm filling out questionnaires. These are very useful and good questionnaires, but there is no biochemical test. And so when we talk about how we can impact, health in a changing environment, we can't just talk about exposures. And so I'll take you very quickly. This is both a scientist's journey and a father's journey on how I want to impact this area, because I just cannot imagine what families are going through without a clear objective measure of conditions like autism spectrum disorder. For those of you who are not familiar with it, the official definition is this: persistent challenges with social communication and interaction, restricted interest, repetitive behavior, there'll be language delays. One thing should shock you about this definition, and that is this: there is no biology in this definition. It's all observational. I'm not saying observation and symptomatology are not important. I'm not saying the social factors are not important. What I am saying is that that cannot be the only thing. That, combined with biology, is a much better definition of this disorder. This is some background. 200 million children are affected globally. There's not a single biochemical test that has been fully approved by the FDA. A condition that is considered heritable is only diagnosed by the age of 4 to 5 in the US, and that's much younger than if you go to Japan or even parts of Europe. They're diagnosing it much later. And if you don't have a biochemical test, then how do you develop a drug? And therefore, there is no FDA-approved drug for the core symptom, the core pathology underlying autism. And all this time, the rates are increasing. Yes, a large part of that increase is better diagnostic and more awareness, but that's not all of it. And this cannot just be genes. Environment must play a role in this, because our genes cannot change this quickly. And all of this increase is in the background of this massive clinical backlog. We don't have enough specialists to diagnose them. Diagnosis takes many hours per individual because we use these lengthy questionnaires, and so they're long wait lists. In many hospitals, I would say A third of hospitals in the US, the wait list is now nudging 1 year or longer just for the diagnosis. So if you can imagine calling a doctor today and saying, I have a concern about autism for my child, and they give you an appointment in the second half of 2027, you will know what I have experienced as a parent. We developed a tool, and I'm not going to go into all the details because I know I've sort of completely shifted from one topic, which was fires and environmental exposures, to this idea of using the same technology but very much in a clinical setting, using environmental exposures as a diagnostic biomarker. So I'll be quick and then I can open this up to questions. But to convince you that this science can be helpful, I want to share the journey at least in a quick way. So this is more than a 10-year journey. I owe a debt of gratitude to so many colleagues who helped me do this study globally in over 2,000 families. So we were part of a national study in Japan where we recruited pregnant women. We collected hair soon after birth, and then the children got the DSM-5 autism diagnosis at the age of 4, 5, and older. I also have colleagues at the Karolinska Institute who helped me study twins, including some of them being identical twins who are discordant for autism. One twin is autistic, the other one does not get a DSM-5 diagnosis of autism. Of course, being at Mount Sinai, we did our own studies, clinical studies here. And when you're developing a clinical tool, you really want to get the FDA to weigh in. They are the gatekeepers of safety and credibility, but also they're excellent at diligence from a clinical perspective. So we submitted all of this to the FDA and we got what's called FDA breakthrough designation. What that means is the FDA says there's no other technology that can do this. You're the first one. We're calling it a breakthrough, but we fast track it then, and you have to work with them to get it to full approval. We also have CLIA certification, which is a FDA mandated process, but it goes state by state. So your laboratory has to meet a certain standard for it to be CLIA certified. And ISO is a global standard. So we got a lot of diligence done, not just peer-reviewed paper, not just NIH grant review, but all of this additional. But this was around 500-600 people. I just wasn't satisfied at this point. So we did additional studies. We took it all the way to a more recent study of almost 1,600 plus families, including a new statewide study in California and a study in a remote, not a remote, but a very impoverished part of Mexico City where environmental exposures are sky high. Honestly, I've never seen lithium exposures this high using the hair biomarker. It was shocking how much lithium there was in these children, and this is not medication, this is just environmental exposure. These results have been under review, and if the editor of Nature Communications is listening, it's been 4 months and I'm still waiting for the first review. So, dude, please hurry up. At least send us a text message or an email. If you're breaking up, let me know, but don't ghost me any longer. I will go quickly through this whole thing. The 580 samples were blinded. We have 96% sensitivity, which helps us being a very good rule-out test. And we have 90% specificity in our high-risk category. We don't rule in. I don't believe anyone should be ruled in for an autism diagnosis based on just a biomarker. It requires clinical expertise. So we say this is a rule-out test. Think of it like Cologuard. It screens you out, but if it cannot screen you out, you go and get that colonoscopy done. This is under review, but this is what it looks like. So no longer does a clinician look at someone who's called them for an autism diagnosis and say, first come, first serve. We can rule out when your risk is low, it is very low. I think we miss 1 in 850 odd cases. So our rule out accuracy is sky high. We can also then triage. We can say, look, these people have a very high risk and they need to be seen immediately. And this is the wait and watch group. So now we are helping various clinics around the country and university hospitals and saying, let us get those people who are on the wrong wait list onto the right one. Autism gets confused for ADHD and other conditions. And often these children wait forever to get an autism evaluation done, only to realize that's not the problem that they are facing. This is my conflict of interest slide where, as you can see, there's company logos, but we have all of this 10 plus years of science gets resolved into one simple box, a tube that you can collect at home. Again, like I said, what I love about this is no needles, no freezing, none of the messiness that comes with a stool test. Rather than end with just a summary slide, I would like to end with more a vision slide. I was talking to the leadership in Washington, the administration, and I said, this is my vision. Why can't we assess children for neurodevelopmental disorders just like we do with neonatal blood spots for metabolic disorders? And those disorders are quite rare. Neurodevelopmental disorders, if you take ADHD and autism together, we're already looking at more than 10%. Mental health is one of the main challenges, main public health challenges of this generation. So what I want to do is run this as a universal screening tool, because you can collect hair as young as 1 month of age. FDA recognizes us for that. And what we realized when we ran an economic model was, because early diagnosis leads to early intervention, which can reduce severity. This is not me saying it. This is many clinical trials showing it, done by others. Overall, it actually saves us money as a healthcare system. So this technology is not meant to just be a way of costing the public health system more. And I'm proud to say that this is something exposomics can achieve. Genomics could not achieve this. I'm not trying to discredit them. They've achieved many other things, but exposomics has achieved this. So we all as environmental scientists should be very proud that exposomics science has now reached a maturity that it can actually help problems, real public health problems, and be incorporated in clinical workflow. This is by no means a singular journey. I'm very grateful to all the people listed here, and especially to the NIH. And among all the NIH funding, I hold a River grant or an R35 grant, which is a unique mechanism, and I would encourage all of you to review similar mechanisms, because it allows you a lot of freedom to do different things. I also want to acknowledge the HERE program, although that has ended. It really moved the needle on the quality of laboratory data that American scientists can access. I'll stop there. Again, thank you so much for the privilege of sharing my work at this symposium, at the seminar series, and I would welcome any questions. Thank you so much.
Thank you. That was just a fascinating talk. I'm always amazed with these talks because it really they're so somewhat outside my my expertise, but really I'm sitting there just calculating different things of how your technique and stuff can be applied to my research in that, and it seems like the sky is the limit to be able to do that. I know we've been doing stuff with hair and stress, picking up the stress, cortisol and stuff like that in it. And now we're having all these, like you were pointing out, that the forest fires and this long-term exposures of all the these fires, and it's not just a one-off anymore. It's multiple times off. It just seems like we have to get unique metrics, and I really think this is very exciting. So, anybody have any questions out there? Go ahead, John.
Kind of held off for a second to see if anybody else wanted to jump in. Can you hear me okay? I got a fan running in the background.
Yep.
Okay.
You're good.
I was wondering if you could talk more about the biomarker that you're looking at. Specifically, what is that biomarker for autism?
Oh, yes. So we published a paper on that. It's not a single marker. It's like a panel, like many genomic panels. This is an environmental panel. And I'll just explain the background so that you get the full picture of how we came up with it. What we found was that in autism, zinc copper cycles play an important role. We have a paper in these junior Nature and Science journals that really explores the role of zinc copper cycles. And this shouldn't surprise any of the clinicians. You would have heard of conditions like Wilson's disease, where you have a genetic disorder that you cannot metabolize copper, and you start having symptoms sometimes that mimic schizophrenia. So zinc copper cycles are well known in neurological disorders. So that was, think of it like a symphony, and zinc copper cycles are the conductor. So that was a core piece, but that's not enough. It doesn't have enough sensitivity and specificity for it to be an autism biomarker. So around that are environmental exposures. Lead is part of it, lithium. Now, if you go to Sweden, lead exposure is not such a big issue. In California, it is somewhat of an issue, and in Mexico, it's a big issue. So surrounding the zinc core of zinc copper cycles, we found that there were metabolic patterns in a bunch of other elements as well. In total, it's a panel of 15 elements, toxic metals, essential nutrients, all of those, that we put together, but we're not analyzing concentration. So no one should walk away thinking, oh, it's because I have high lead exposure. It's more like I cannot metabolize lead exposure as well as a non-autistic person. So it's the difference in the metabolism of that. And so that's why the time dimension and having so many, such tense temporal resolution becomes very important. I hope I've answered your question, John.
Yeah, sorry, just real quick. First of all, I should have led with just saying that was a great talk. It was really
too late, John. Now I'm already devastated. I'm going to drown my sorrows in another coffee.
But no, so I guess what I'm hearing is kind of a fingerprint of a number of different markers that you're looking at, and that fingerprint may differ depending on whether you're looking at wildfire smoke or autism.
Exactly.
The fingerprint, exactly. I guess, is pretty distinctly different, I guess, between those.
You've nailed it, and I describe it in a similar way, but back in the day, there was this app called Shazam. You would hold up your phone and open up the Shazam app. If you were listening to some music and you couldn't remember the song, it would tell you what song it is. Now, a Google app does it for you as well. And all it was doing was looking at this temporal pattern in the background. In fact, we use similar mathematical or modeling techniques to that app. And we're just modeling these patterns over time and saying, oh, this pattern just seems to be very sensitive and specific to autism because we have compared it to ADHD, neurotypical kids with just anxiety or language delay. And these other patterns are because the exposure is happening and we are breathing it in. And so we are just doing pattern recognition, but at a molecular level. So it's like your ECG, but instead of looking at one trace, we are looking at 10,000 traces.
Anyone else have any questions? If not, thank you very much for your time and presenting. Like I said, I found it very fascinating and everything. So I appreciate your time. And yeah, I think there might be a few of us that will probably be reaching out for potential collaborations even.
Absolutely. I'm always happy to help. And thank you again for the privilege of being here. I really appreciate it. All the best, everyone. Bye-bye.
All right. Thank you.